Imagine a world where the tiny organisms living inside us could predict how well we fight cancer. Sounds like science fiction, right? But groundbreaking research is revealing that the microbiome, the vast community of microbes in our bodies, might hold the key to understanding and treating clear cell renal cell carcinoma, ccRCC, a deadly kidney cancer with a staggering 40% post-surgery recurrence rate. This aggressive disease often requires radical surgery, targeted therapies, and immune checkpoint inhibitors, yet outcomes remain uncertain. Here's the part most people miss. Current treatments and molecular subtyping methods focus solely on human genes, completely ignoring the microbiome's potential role. But here's where it gets controversial. Could these microscopic inhabitants of our bodies be silent players in cancer's progression, or even its cure? Recent studies hint that microbiome imbalances might fuel diseases like cancer, but most research relies on gut samples, potentially overlooking organ-specific microbes. Enter next-generation sequencing, which has uncovered bacterial traces in tumor tissues, including those from the Cancer Genome Atlas (TCGA) project. This suggests the microbiome could revolutionize cancer diagnostics and prognostics. Led by Xingang from Shanghai Jiaotong University and Brian Rini from Vanderbilt University, a team of researchers dove deep into the ccRCC tumor microbiome, linking it to patient outcomes and molecular traits. By integrating microbiome, metabolome, and transcriptome data from multiple centers, they've opened a new frontier in precision medicine for this disease. The team analyzed tumor and adjacent healthy tissues from untreated ccRCC patients using 16S rRNA sequencing and untargeted metabolomics. After filtering out low-quality samples, they found tumor tissues had significantly higher microbial diversity compared to healthy tissues, as measured by indices like ACE, CHAR1, and Shannon. Interestingly, aerobic bacteria were less abundant in tumors, while anaerobic and facultative anaerobes remained stable, a finding that aligns with ccRCC's oxygen-starved environment. Volcano plots and RNA fluorescence in situ hybridization, FISH, confirmed bacterial presence in tumors, pinpointing specific microbial culprits. Using TCGA data from 428 patients, the researchers classified ccRCC into 3 microbiome-based subtypes using algorithms like int-NMF and SNF. Survival analysis revealed stark differences. Group 1 and Group 3 had poorer outcomes compared to Group 2. Gene set enrichment analysis, GSEA, showed Group 1 had overactive metabolic pathways but suppressed immune responses, while Group 3 lacked anti-tumor immunity and displayed unusual metabolic traits. Whole exome sequencing uncovered distinct mutation patterns. Group 2 had fewer VHL and PBRM1 mutations, while SETD2 and BAP1 mutations were common across all groups, linking them to metabolic and immune dysfunction. Metabolomic analysis added another layer, showing glycerophospholipid metabolism was heightened in Groups 1 and 2. While linoleic acid metabolism was elevated in Groups 1 and 3. Though preliminary, abnormal linoleic acid metabolism might signal poor prognosis, warranting further study. This study not only categorizes ccRCC into 3 microbiome-driven subtypes but also highlights the microbiome's role as an independent prognostic marker, complementing human genome data. It paves the way for personalized treatments and risk stratification. However, the small sample size and incomplete data matching are limitations that call for larger studies. But here's the burning question. Could manipulating the microbiome become a game-changer in cancer therapy? While this research is in its early stages, it challenges us to rethink cancer's complexity and invites debate on the microbiome's untapped potential. What do you think? Could microbes hold the key to beating cancer? Or is this just another scientific detour? Share your thoughts below. Let's spark a conversation.